Hello everyone. Here we'll try to learn the phototransduction in the rods and cones, for which we are drawing here the outer and the inner segment of the rod and cones. This is the inner this is the inner segment and this is the outer segment of the cone or rod, and this is the one disc that which is seen in the rod, and we can see the photoreceptor. So this is the rhodopsin, and this is the transducin, the GTP, and this is the phosphodiesterase enzyme, and this is the rod or cone. So this is the outer segment. So the mitochondria, this is nucleus, and this is the rod or cone. So this is the inhibitory neuron, this is the bipolar neuron, and this is ganglion cell. When there is no light, the GTP under the influence of guanyl cyclase normally gets converted into cyclic GMP. And this cyclic GMP, higher levels of cyclic GMP keeps the sodium channels open and the sodium influx occurs. That causes depolarization, and this depolarization in the lower segment leads to the opening of the calcium channel. So calcium causes the degranulation of the glutamate vesicles. Release of glutamate leads to activation of inhibitory cells here, and that causes inhibition at the bipolar cell. And when there is when light falls on the photopigment, that is retinin gets converted from the cis retinal to trans retinal. That is going to activate the transducin proteins, that is GT1 and 2, GT1 in rods and GT2 in cones. So that in turn is going to convert the cyclic GMP into 5-GMP. So that causes the decreased levels of cyclic GMP causes the closure of the sodium channels. So that leads to hyperpolarization, and hyperpolarization leads to decreased release in the glutamate. That is going to inhibit the inhibitory neurons, and inhibitory neurons in turn bipolar. That this disinhibition causes excitation of the polar cell and is going to send impulses to the ganglion, to the brain,